I will explain how this bioinformatics is important in different complexity of living systems. So, if you talk about the complexity of biological systems, okay, so we have the different ways. First we start with DNA nucleic acid sequence, right, I put a nucleic acid sequence here, right. So, then we developed with the nucleotide structure. So, if we started with the what are different nucleotide bases A, T, C, G, right. So, if we go with the nucleotide sequence, then we go with the nucleotide structure, and from nucleotide structure then we go with the gene expression. So, we go with the DNA, DNA to RNA and RNA to protein, right. So, we can go to the gene expression and finally, we get the protein sequence. So, once the protein sequence, then we go over to the next level, this is called protein structure, and then from protein structure to protein interactions, and protein interactions, then this is happen in the cell, right, and then we go for the cell signaling and go with the tissues and develop the organs and the physiology of the system and finally the organism. So, this is the complexity of the biological systems. So, how bioinformatics contribute to understand the complexity of biological systems. So, if you look into the applications of bioinformatics, you can see there are various aspects to contribute in different stages of these biological systems. For example, if you go to the nucleotide sequence, how bioinformatics can contribute to nucleotide sequence? Storage of large number of sequences and analyze these nucleotide sequences, right. So, for example, if you look into this nucleotide sequence, there are millions of sequences are available now. So, they are stored in the kind of databases. For example, you can say the few types of databases. Can you list the name of few databases? NCBI. So, GenBank you can get, right, EMBL you have the data, and mainly the DNA data bank of Japan, DDBJ, right. So, there are various databases. So, they collect the data, right, put everything together and they organize in a specific way, right. All these databases are freely available and you can have several search options to extract the data and you can use this information for analysis. The first one is we have the clear database for the nucleic acid sequence, and when this database is ready, then we can do the analysis, right. Which type of analysis we can do from the nucleic acid sequence? You can see interaction analysis, you can see AT content, or you can see GC content, okay, are there any specific properties. For example, whether the DNA regions are highly bending or is highly flexible, because flexibility is important for the binding, right. So, you can derive various features. For example, if you have a DNA sequence, what is the melting temperature? This can withstand for high temperature or low temperature. So, all the information we can get from the bioinformatics analysis. Now, we go with the nucleic acid sequence to nucleic acid structure, right. What are the information you can how bioinformatics contribute to the nucleic acid structure? So, if you have the structures, right, first you can make a database, right. For can you mention one database for nucleic acid structure? Yeah, PDB contains the data, the PDB is protein data bank anyway. So, nucleic acid database also contains the information regarding nucleotide structure. So, then you can also, as you mentioned, you can also predict the structure from the sequence, that there are many bioinformatics tools available to predict the nucleotide structure from the sequence. Then from the structures you can analyze various parameters, for example, various base step parameters for the nucleotides, right. How about the base stacking energy of the different bases, how is the base pairing energy, how about the hydrogen bonds, right, various types of interactions. So, all this information you can get from the nucleotide structures, and we have different algorithms available in the literature to analyze the nucleic acid structures as well as to predict the nucleotide structures from the nucleic acid sequence. Then go with the next step, right, from the nucleotide structure to the gene expression. So, how the bioinformatics contribute to gene expression?
You can predict the gene expression using bioinformatics, means how many proteins, how many RNA sequences are transcribed from the gene.
Correct. So, you can have the coding regions, you can get uncoding regions, and you can translate the sequences, the DNA sequences to the protein sequence and so on, right. So, you can have the algorithms to convert this, translate these DNA sequences to protein sequences and so on. Then go with the protein sequence. So, how the bioinformatics help in protein sequence?
From the gene expression, it can again predict the protein sequence, what protein is probably made. And from protein sequence, it can identify what is the contribution of each residue in different kinds of proteins. So
correct. So, if you go over the first protein sequence, right. So, many sequences which are deposited, right, are accumulated and they are curated and deposited in the form of a database, right. Can you tell the database which contains the protein sequences? UniProt, right. UniProt is a database, right. So, UniProt is a database which contains protein sequence information, right. So, earlier it was started with the PIR, protein information resource, right. In the meantime, the Swissprot also developed a database. They merged together, right, 10 to 15 years ago. So they made a combined effort of a consortium called the UniProt consortium. So they collect all the information regarding protein sequences and they put together in the form of a database called UniProt database. Currently, how many sequences are known in UniProt database? 17 million sequences, right. So currently, if you see the UniProt contains two types of databases. One is the computationally annotated and the second one is the manually curated. So they have the manually curated database and the computer annotated. Right. So currently 55,000 sequences from the manually curated and the 78 million sequences using computer annotated sequences, right. So total you had about 78 million sequences, right. So you have the bioinformatics database, right, which contains all the information. This database is widely used in the literature because this database contains lot of information, right. What are information you can obtain from UniProt database? You can see the function notation, links to the structures, interactions with other proteins, linking mutation diseases, post translational modifications, isoforms, amino acid sequences in different formats, in FASTA format and the different format styles. So it starts with the protein names and the synonymous and what are the various sources and organisms, and finally go with the functions, right, and they give almost all the information regarding the particular protein, as well as they give the literature and linked with the other databases. So it is high integrated ones, less redundancy and highly reliable, right, highly annotated. So there is a reason why the UniProt is the unique resource for any protein sequences, because it contains protein sequence database, it has all the information regarding a particular protein. So if you are interested in any particular protein, you can go to UniProt, right, and you can use it to get the information regarding your particular protein. Right. Also it has high level of annotations and linked with the other databases, right. So then we can use the information available in the UniProt. So if you go here, so you have the protein sequence, now you have the database, right, 70 million sequences. Now what can we do with these sequences? We can also use the sequences to extract information. So what are the various informations you can extract from the protein sequences? So now if you have the protein sequence, right, what are the various information you obtain from the protein sequence using bioinformatics? You can calculate the composition, right. What is a composition?
Number of amino acids.
Okay, so I will explain the details in later classes, just I will have a brief explanation I will give you now. So if you have a sequence, for example if you have this sequence, right. So there is another sequence you can see like this. So if you look at the sequences, there are some specific preference of amino acid residues, right. If you see the first sequence, which residue is highly preferred? Can you see how many times A occurs? 2 times, I 3 times, T 3 times, right. So if we see this sequence, so how many times A occurs? 5 times, I 0, T 4 times, right, 1 2 3, this is T, then this is 5, right, 5 times, right. So now if you see the different sequences, you can see the difference. Some residues are preferred in some sequences, some residues not preferred in some sequences, right. So if we have the sequences for several types of proteins, so you can see the number of times each amino acid residue occur, right, and you can see this has some bias in the protein structures. If we got different types of proteins, for example DNA binding proteins, you know DNA binding proteins, the protein interact with the DNA. So DNA is the negatively charged because of phosphate group. So here we have the binding region, you can see the preference of positive charge residues, right. What are the positive charge residues? Arginine, lysine, right, you can see the preference of positive charge residues. So if you have any small stretch of regions with the occurrence of more number of positive charge residues, then you can see that this sequence could be a DNA or RNA binding protein, right. Likewise, if any protein contains a stretch of hydrophobic residues, for example more than 15 to 16 hydrophobic residues, then we can see that this protein that sequence could probably be a transmembrane helical proteins, because in the transmembrane helical proteins you can have a stretch of hydrophobic residues inside the membrane, right. So, for you see the helical proteins, so there is a membrane, right, that this is the helical regions here, this is highly dominated by the hydrophobic residues, right. So, you can see that. So, likewise if you have a sequence in the uniprot, so you can extract various information, right. So, this occurrence, now here in this occurrence the numbers depend on the length, right. For example, if you have 100 residues and another protein and 1000 residues, then if you see the occurrence, right. So, A will be around 10 or D will be around 10 or so on. If you have the 1000 residue protein, you probably divided multiplied by 20, right, randomly if it is distributed, right, how many residues in this protein, right. For example, if you have 100 residues, the protein contains 100 residues, if all the 20 residues are randomly distributed, right, what is the occurrence of a particular residue? 5, right. On the other hand, if there are 1000 residues, so n of A equal to 50, right. So, the length matters, if you have different proteins at different lengths, the number will be different. So, comparing these two, we cannot say that this A is highly dominant in the case of the second protein. So, in this case we need to normalize, right. How to normalize it? With length. So, in this case composition is the occurrence divided by length. In this case if you have 100 residues, right, randomly distributed, right, what is the composition? This is 5 by 100, right, this equal to 0.05. For the second case, right, you have 1000 residues. So, what is the composition for alanine? 50 by 1000, right, this equal to 0.05, right. So, in this case if you see, if you look into this just the sequence length, so you can see a difference, but if you normalize with the length, so there is same. In this case even it is 100 residue protein or 1000 residue protein, so the occurrence, the composition is the same, right. In this case there is no bias, right. This is the reason why we need to normalize with the chain length to get the composition. So, look into this UniProt database, there are 78 million sequences and you try to obtain this composition for 20 different amino acid residues, right, you can see some sort of bias, right, for all 20 amino acid residues. If all the 20 residues are equally distributed, right, then what is the percentage of each residue is 5%, right, if they are randomly distributed. But we look into this UniProt database and the sequences, there is not the same, right, some residues are higher occurrence and some are less, right. Can you tell some which residues are highly dominant and which residues are less preference?
Tryptophan is very less preferred.
Yeah, tryptophan and cysteine, these residues are less preferred, and if you see the hydrophobic residues, right, valine or alanine, right, you can say highly preferred, they have more than about 10%, right. In the case of cysteine or the tryptophan it is about 2 to 3%, right, because nature selects in such a way that the proteins from the unfolded state to the folded state, they because of hydrophobic collapse, right, these residues come close to each other. So, there is dominance of the hydrophobic residues, and then once they are done, then there is maintained the stability by other interactions like the hydrogen bonding and electrostatic interactions, right. So, you can see the threonine and the serine and the charge residues, they are next level of preference in the whole amino acid sequence. So, if you have the sequence, the bioinformatics contribute to various levels to understand the different types of proteins based on the structure and based on the function, right. So, we discussed about two types of the features, occurrence or the composition. There are various features you can derive. For example, you can construct some profiles for properties, you can get the average property of the particular sequence and you can see whether any specific residue is conserved. That means, so if you have a sequence, right. So, you see several organisms, several species, you check the data and see there any particular residue which is present in all the places, in all the organisms. If it is present in all the organisms, then you can say that this particular residue is highly conserved and this plays an important role in the structure and function, and if you disturb this residue, that will create several problems in the particular protein and cause some diseases, right, probably. So, in this case you can see this information from the sequences, because a lot of sequence available. So, take any sequence, make similar sequences together and see any residue is present at the same position in all the organisms and how they are important for the function. So, protein sequence you can see the application of bioinformatics to derive the various features of the protein sequence. Okay, then there are various other factors, so that I will explain later, right, when the future classes. Then next level we go to the protein structure, right, how the bioinformatics contribute to protein structure.
Protein structure prediction can be done. Apart from that, structure can be used in various functions. For example, in enzymes, the interaction pattern analysis and allosteric catalytic sites.
So, catalytic sites, yeah, right. So, you have lot of ways you can use the protein structures using bioinformatics. First, we try to have a database, right, which is database which contains protein structures, protein data bank, right. Protein data bank is a unique resource, right, for obtaining protein structures, right. So, currently how many structures are deposited in protein data bank? Approximately 130,000 structures are deposited in the protein data bank. So, we have the protein structures, right, and how many sequences are deposited in the UniProt database? 78 million sequences, right. So, if you see the difference of known information between the sequences and the structures, there is about 600 to 700 fold. So, mainly the structure determines the function. So, in this case it is very important to get the structure for any sequence if the structure is not known, right, that is called a protein folding problem. So, predict the structure of a protein from the sequence, because since sequences are known, right, for 78 million and the structures are known very less. So, you can have a relationship. When you determine the structures, it takes time, right, it also involves lot of manpower and the time and the money, right, it is cost effect. So, we try to predict. So, using bioinformatics tools we can predict the structure of any protein from its sequence. So, we have a sequence we can try to predict, right. The accuracy levels are about it's about 80% now. So, in later classes I will explain how to predict the structure from a sequence. There are various methods available to predict the structure, right. The foremost one is homology modeling, because if two sequences are similar, then we assume that the structures are similar, right. Using this concept, right, you can derive say methods based on homology modeling to predict the structure from the amino acid sequence. And you can do the fold recognition, because if you have any specific fold, you can use the fold recognition technique, and you can use threading as well as you can use the ab initio method. The ab initio modeling, so we start from the scratch, right, starting from the bond length, bond angle, torsion angle, right, and finally we use calculate the energy and the energy minimization techniques to predict the 3D structures. The only the disadvantage of this ab initio modeling is it can handle some limited number of residues, because it takes enormous computational power and the time. In this case they cut into pieces and then do the modeling and they join together, and finally they minimize the energy to get the final folded structure. So, now if we have the structure, we can predict the structure from the sequence, and if we have the structures, what are the various information you can obtain? You can use the bioinformatics tools, right, or you can use the bioinformatics algorithms to derive various features from the structures, right. For example, what are the various features you can derive from the protein structures? Solid accessibility you can do. For example, if you have a protein, right, so if you have a protein here, I showed a protein. So, some residues which are here, right. So, these residues are not easily accessible. So, if there are some there in the outer surface, if you see here, so these residues are highly accessible. So, if we have the structure, we can easily see which residues are buried, that is inside the core, and which residues are at the surface. Why do you need that information? Yeah, we can see, if you from this you can see which residues can interact with other systems, other proteins or ligands or nucleic acids and so on. If there is at the interior core, they are responsible mainly for keeping the protein stable, right, they are mainly contributing to the folding and stability. So, if you look into these residues which are at the outer surface, right, these residues they tend to interact with other molecules such as proteins or nucleic acids or small molecules, right, for the interactions, right, and for the function. And this is also important for the structure based drug design, just I discussed earlier. So, if you have a target and to identify the ligands to interact, so that should have some pockets, right. If you know the structure, you can identify the binding pockets, the binding sites where a small molecule or a ligand can interact with the amino acid residues in a protein. And if you have 3D structures, right, there are various many residues in a sequence, and these residues interact each other and form the structure, right. This can easily tell you which residues are close to each other. So, for example, if you have the 3D structures, right, this is a sequence, they form a 3D structure like this. There are various occasions where these two residues which are far away in the sequence, how many residues far apart? 11 residues far apart, right, but it can be possible these residues are close to each other. So, for example, this A is here, A1 is here and the I12 is here, right. So, if we have the 3D structures, you can get this information where how these residues interact with each other and how they are located, right. So, in this case you can see the contacts, where this is amino acid sequence, here also amino acid sequence, and if this residue number 1, 2, 3, 4, 5 like this, right. So, how whether they are close or they are far. In this case you can see, for example, if this and this are very far, they can be close in space. Some cases the close residues they are always close. So, you can see the information that how far the residues in the amino acid sequence are distributed very far away and the close are come together in the case of protein 3D structures, right. How the residues in the space are distributed in the sequence, right, that you get the information like that. Then you can see the residues which contribute from far away and which residues make a short range contacts, and all the information you get from the 3D structures. So, now if you have the structures, then also you can derive various features, right. For example, you can see the contact order, long range order and the hydrophobic behavior of residues and various aspects you can obtain from the 3D structures. Then we translate this information to predict the structures to understand various functions of these proteins, right. This is the reason why we try to get several parameters from protein structures. So, now next level we got the protein interactions. What are the various protein interactions? Protein nucleic acid interactions, protein small molecule interactions, protein interactions, right. So, now in the protein interactions also bioinformatics contributed significantly to identify the residues, right. So, because crystallographers they tried solve the structures and then deposit the structure, that is it. So, to understand the residues and you have the uniform information where the proteins can interact and which residues prefer to interact and why these residues are prefer to interact, right, or any specific patterns available for two proteins to interact. So, in this case we use bioinformatics to understand these specific binding sites, right. The various criteria to define the binding sites, right. What are the various criteria used to define the binding sites? You can use a distance, right, if they are very close to each other then these proteins try to interact, and you can use the acceptable surface area, right, how far the surface area reduced upon binding. So, based on that you can see whether any these residues interact or not. You can calculate the interaction energy, right, and from that you can define whether these two proteins interact or not. Likewise, you can see the protein ligand interactions, protein RNA interactions, protein DNA interactions, all these you can do. Then there are various databases available for protein interactions, right, on various aspects. First one is you can see the interacting patterns, interacting pairs, which two proteins interact, right, and the interacting sites, right, and specific to some of the genomes like human genome. If you take the human genome, currently how many proteins in human genome? How many proteins? Yeah, 20,000 plus, right. So, earlier it was assumed to be 100,000, then 30,000, 50,000, right. Currently the UniProt, if you see around 20,000 plus sequences in the UniProt database, right. So, if all these sequences, then which proteins interact with each other? What are the two pairs interact, right. So, this information we collected the information, there are various database for interacting proteins, right, and for the binding affinity of these proteins. If the protein A interacts with protein B, what is the binding affinity of these two proteins to interact. So, there are various databases available for the case of the protein interactions, right. So, from these interaction studies we can try to derive the hypothesis how the two proteins interact with each other, right. Likewise, we can see the other aspect like cell, right. You can have the various databases as well as analysis, right, to understand the different activities of the cell and cell signaling and different pathways, right, and have the tissue data to see the organs and physiology as well as the organisms, right, for example neuroscience and so on, right. So, you can use the bioinformatics to understand the complexity of the various levels of complexities in biological systems, right. In the next classes I will start with the different aspects starting with nucleotide sequence to up to these different types of interactions.